Hello each and everyone, welcome back to my channel ANAMIKA'S EDUSPACE, and hi, this is Nida Mishra. So with me we are going to learn glycolysis. So let's begin, let's dive in. And so the first step of glycolysis, we all know that we are going to start with glucose, that is a 6-carbon compound, and we are going to break down glucose into pyruvate. So the first step where glucose is converted into glucose 6-phosphate with the help of the enzyme that is hexokinase. That means a phosphate group is added, and with the 2 sugars we can actually begin the first step of glycolysis. That means if it is a glucose sugar, that is a simple sugar, we can convert it into pyruvate, or if it is a fructose sugar, we can also convert it into pyruvate. So first step will be, that is glucose is converted into glucose 6-phosphate, and here we are providing the ATP. That means here energy is provided into the steps and we are getting or gaining ADP out of it. That means 1 phosphate has been already given to the glucose, and that's why in the 6-carbon compound the phosphate is added. That's why the name comes that glucose 6-phosphate. Okay, in this way if we study, then we will learn it very quickly if we understand it. And also very important, the enzyme, and the enzyme question definitely comes in the examination, that what type of enzyme is used in this process, and that is nothing other than hexokinase, because hexokinase will ultimately help to break that bond and add on the phosphate group to the carbon, right? 6th carbon of the glucose. Okay, now coming to the next step. So similarly we will go on through the isomerism process. So isomerism process is just that same sugar or same structural formula will be there. That means glucose and fructose are the same structural sugars, as we can see, right? So from glucose 6-phosphate we are converting it into fructose 6-phosphate. Also fructose can enter the glycolysis cycle from the other hand, and that's why at the right hand side of the cycle we can also write fructose, right? Again, next, after converting the sugar, we are going to convert the fructose 6-phosphate into fructose 1,6-bisphosphate with the help of the enzyme that is phosphofructokinase. So again, whenever we will hear about the enzyme kinase, that means any kinases or any division is going on, and at that point a new phosphate or a new energy compound is added, children. So that's the thing. The first 6-carbon compound, the phosphate group was added, and now again at the 1st carbon compound the phosphate group is added. Now if the question comes that why at the first and why again at the 6th, that is also an analogy which is going on inside, which we will learn in the higher classes or in the detailed video of it. Now let's carry on again with the cycle. So now we are going to learn about the next step of the glucose, that is when fructose 1,6-bisphosphate is divided into 2 main steps. So fructose 1,6-bisphosphate will be ultimately divided into 2 steps. One is dihydroxyacetone phosphate, and another it will be divided into glyceraldehyde 3-phosphate, right? And this division, in this division to another thing will be asked, that which enzyme is used in this division. So that enzyme will be aldolase. So that enzyme will be none other than aldolase, which is actually doing the cleavage, and dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, both are the 2-carbon compounds which are unstable in nature. That means one time it can turn into dihydroxyacetone phosphate, another time it can turn into glyceraldehyde 3-phosphate. Okay, now we will carry on the step with the glyceraldehyde 3-phosphate, because glyceraldehyde 3-phosphate is a little bit more stability, gaining more stability. So we will carry on with glyceraldehyde 3-phosphate, leaving dihydroxyacetone phosphate aside. Okay, next we will use the next enzyme, that is triose phosphate isomerase, and in this way dihydroxyacetone phosphate and glyceraldehyde 3-phosphate will be keeping a balance in between each other. We are going to convert the glyceraldehyde 3-phosphate with the help of glyceraldehyde 3-phosphate dehydrogenase, and here remember, when dehydrogenase enzyme is used, that means the compound is giving one hydrogen group going for the reduction, and here also 3-phosphate, that means phosphate was added on the 3-carbon compound. Now the phosphate is added on the 1st carbon compound as well. So the ultimate step or ultimate substrate we get, that is 1,3-bisphosphate glycerate, right? After 1,3-bisphosphate glycerate, we get another step, that is 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate with the first rate determining step, and this is also a energy gaining step. So this is the first ATP synthesis of the whole glycolysis. And also children, do remember that in glycolysis we only gain ATP, that is 2 ATP we gain, and that is also 2 times. One time, this is the furthest in this step we are going to get the ATP first when 1,3-bis-phosphoglycerate is converted into 3-phosphoglycerate. At that time we are going to gain the ATP. That means ADP will be given to the reaction, and ADP will be turning into ATP in this reaction with the help of the enzyme, that is phosphoglycerate kinase. Right? Again, kinase is there, that means one phosphate group is moving out. Now see, the next step is very easy, where you have written 3-phosphoglycerate. 3-phosphoglycerate is going to transform into 2-phosphoglycerate with the help of the enzyme mutase. Phosphoglycerate mutase. Right? So mutation, you all know that mutation means any change in the sequence, any change in the format, right? Or any change in the orientation. Similarly, 3-phosphoglycerate is converted into 2-phosphoglycerate with the help of the enzyme mutase, right? Here also we are, actually the phosphate group is moving from the 3-carbon compound to the 2-carbon compound. Now we are moving to the next stage. We are going to add an enol into it with the help of the enzyme, that is enolase. So the last substrate which we have got, that is 2-phosphoglycerate. 2-phosphoglycerate is going to convert into phosphoenol pyruvate. Here actually the phosphate group activates, and it actually attracts the enol compound into it, and ultimately it turns into phosphoenol pyruvate. So I think I am clear to the second last step of glycolysis. Now we are heading to the last step of glycolysis, that is phosphoenol pyruvate will be transformed into pyruvate with the gaining of another ATP molecule. That means here also in the reaction we are providing ADP, and the phospho group is getting detached from the pyruvate molecule and leaving enolase as the enzyme. And here which enzyme is working? Pyruvate kinase is working. It is again breaking down. We are getting the phosphate compound aside. ADP is turning into ATP, that means addition of the phosphate group to the ATP molecule, right? And ultimately pyruvate, that is a 2-carbon compound, is produced. So our main aim was to break down the carbon chain or the long chain of glucose, right? So glucose was a 6-carbon compound, and now we get pyruvate, which is a carbon compound. Now pyruvate, up till this, from glucose to pyruvate, this 10 steps of glycolysis, we will find only 3 irreversible states. So the first step, which is irreversible, that is glucose to glucose 6-phosphate, is irreversible step. Next step which is irreversible, fructose 6-phosphate to the fructose 1,6-bisphosphate, right? And the third step which is irreversible, that is the last step of glycolysis, where phosphoenol pyruvate is converted into pyruvate. So up till this, it was all about the common pathway for both aerobic and anaerobic. After this, the pyruvate will be the common substrate, and after this it will go on with oxygen and without oxygen. So when it will go with oxygen, it will carry on through the aerobic cycle, and when it will go without oxygen, it will carry on through the anaerobic cycle. So this is the whole step of glycolysis. Yes, you can also wait for the next mnemonics in my session. Till then, stay tuned. Okay, goodbye.